Today we are going to learn about the interactions between acids and bases within the human gastrointestinal system, and primarily their function in buffer solutions to prevent pH rising and pH decreasing upon the consumption of food. But before we can learn about such complex interactions, we must first learn about the basic principles of acids and bases. We know that acids and bases are an integral part of chemistry, but what exactly do they do, and how do they work? To put it simply, acids are molecules which when in solution have a pH below 7, while bases are molecules which when in solution have a pH above 7.
pH is merely a means of logarithmically defining the concentration of H3O+ ions in any given solution. Many common acids are found in foods, while many common bases are found in cleaning items. Acids and bases are used in buffer solutions to regulate pH within a certain range. This regulation is necessary in order to maximize efficiency of biological functions. One of the most common examples of the importance of pH regulation is the denaturing of enzymes through citric acid to prevent browning of an apple.
But what exactly is a buffer solution? Well, a buffer solution contains both an acid and the acid's conjugate base. An equilibrium eventually will take form, and this equilibrium allows the buffer to resist small changes in pH by shifting the reaction towards either the products or reactant side with respect to whatever molecule is introduced into the solution. So essentially, when too much citric acid is added to an apple, the buffer solution is unable to properly regulate the pH, and as a result, the enzymes responsible for apple browning become denatured and unable to function.
A highly varied range of pHs occur throughout the body, but the most important functions of pH and buffers occur in the digestive and circulatory system. We will first start with the digestive system. The main goal of the digestive system is to break down ingested foods into molecules that can be easily absorbed and used by the body to build and maintain biological processes. The first major example of acids and bases in the digestive system occurs in the stomach. Hydrochloric acid is a key player in gastric juice, as shown in the figure. The cells lining the stomach excrete hydrogen and chloride ions. This powerful gastric juice produces the lowest pH of any solution in the body, between 1.5 and 3.5. The primary function of the gastric juice is to denature and break up proteins into smaller pieces so that the food can be easily absorbed later on. In response to food entering the stomach, secretion greatly increases within 90 minutes of ingestion. The cells of the stomach lining are highly basic to prevent the gastric juice from eating away at the stomach. However, the hydrochloric acid can only denature proteins so much. Help comes in the form of pepsinogen, an enzyme that, once activated, is capable of breaking down proteins. It is released from the cell wall of the stomach and is activated by the HCl within the gastric juices, resulting in becoming pepsin. The pepsin is then capable of further denaturing the proteins to become smaller peptides, and further digestion of the food is able to begin within the small intestine.
So now, most of the proteins are denatured and ready to be absorbed. The highly acidic solution now moves from the stomach into the main center of digestion and absorption, known as the duodenum of the small intestine. In order for nutrient absorption within the small intestine to begin, a buffer solution must take place for the neutralization of residual hydrochloric acid. Without this neutralization taking place, the natural pH ranges of the tissues following the stomach would be disrupted. The duodenum, jejunum, and ileum of the small intestine have pre-ingestion pH ranges of 6.4, 6.6, and 7.4, respectively. A sodium bicarbonate solution, with a pH of roughly 8.8, is released from the pancreas and flows into the duodenum at a rate almost matching the secretion of hydrochloric acid in the stomach. Sodium carbonate and hydrochloric acid react completely to form sodium chloride and carbonic acid. Excess sodium chloride is expelled through the excretory system, and carbonic acid is absorbed into the bloodstream, where the most complex acid-base interactions occur.
Another important buffer exists in the blood as well, in order to tightly maintain the pH around 7.4. Carbonic acid, formed as a result of sodium carbonate reacting with HCl in the digestive system, and a reaction between dissolved carbon dioxide and water in blood plasma, is the main buffer that exists in blood. The components of carbonic acid exist in an aqueous double equilibrium, of dissolved CO2 and water, carbonic acid, and its associated dissolved ions, H plus and HCO3. The conjugate base of carbonic acid, HCO3, acts as the weak base in this buffer. As metabolic acids are produced as a result of regular cellular processes and the absorption of other associated chemicals, H plus ions are released into the bloodstream, and as a result, H3O molecules are formed. Thus, the equilibrium shifts left to form complete molecules of carbonic acid. Carbonic acid also acts as an important carrier of CO2 molecules. As blood approaches the alveoli, the carbonic acid decomposes and releases gaseous CO2 into the lungs in order to be expelled by exhalation.
To recap, the body is highly reliant on acids and bases in the absorption of food, especially through the highly acidic environment of the stomach. The buffers of sodium carbonate in the small intestine and carbonic acid in the blood shift equilibrium in order to tightly regulate pH and maintain maximum efficiency of body tissues.